The mitochondria might be the powerhouse of the cell, but we make ATP before we even get there, and we do that through this process called substrate-level phosphorylation. So starting with the word substrate, we know that a substrate is a molecule that binds to an enzyme at its active site, and that's going to allow that enzyme to catalyze a reaction. So when we have a substrate and an enzyme bound to each other, it's called the enzyme-substrate complex. So then we have the word phosphorylation. If we break down that word, we get phospho, which refers to a phosphate group, and then we get ylation, which is a suffix meaning the process of adding a functional group. So phosphorylation, it's the process of adding a phosphate group to a molecule. But what exactly is SLP? It is one of the ways that our cells are going to directly make ATP during cellular respiration. But there's a catch. Only glycolysis and the citric acid cycle are going to use this method to make ATP. Oxidative phosphorylation is going to use a method called chemiosmosis. So essentially, SLP is going to allow ATP to be made without needing oxygen or even the mitochondria. It's going to work by transferring a phosphate group from a high-energy molecule directly to ADP by using an enzyme. And here's the key point. The high-energy molecules in substrate-level phosphorylation, they are not our electron carriers like NADH or FADH2. Those high-energy molecules, they are different from our electron carriers. So how exactly does substrate-level phosphorylation work? So whether we are in glycolysis or the citric acid cycle, the steps are going to be generally the same, but we're going to start with glycolysis. So to begin, this is a phosphate group. It's a central phosphorus atom that is double bonded to one oxygen and then single bonded to two other oxygens. That R group you see, it's attached and it's going to represent whatever molecule is bonded to the phosphate. So in glycolysis, we have two high energy molecules. We have 1,3-bisphosphoglycerate, which is 1,3-BPG for short, or we have phosphoenol pyruvate, which is PEP for short. And the high energy molecule that we use is going to depend on which step of glycolysis we are in. So glycolysis, it takes place in the cytosol and it has two phases. It has the energy investment phase in which we're going to use 2-ATP to convert glucose into 2-glyceraldehyde 3-phosphate, which is G3P for short. Then we have the energy payoff phase. So from G3P forward, everything is going to happen 2 times per glucose molecule. So at step 7, the enzyme phosphoglycerate kinase is going to take our high energy molecule 1,3-BPG and convert it into 3-phosphoglycerate. This step is substrate level phosphorylation and it produces 1 ATP. A couple of steps later, we reach phosphoenol pyruvate, our other high energy molecule. That PEP is going to be converted into pyruvate by pyruvate kinases. This is going to be another substrate level phosphorylation step. It's also going to make 1 ATP. But because each of these is going to happen 2 times for glucose, then glycolysis is going to make a total of 4 ATP through substrate level phosphorylation. But we are only going to have a net gain of 2 because we invested 2 earlier in the cycle. So looking at the diagram, we're going to look at the enzyme substrate complex that we have in SLP and glycolysis. In each SLP step, the enzyme that we have, which in glycolysis can either be phosphoglycerate kinase or pyruvate kinase, it's going to bind to 2 substrates. It's going to bind to the high energy molecule, which we either have 1,3-BPG or PEP, and it's also going to bind to ADP. Once the enzyme binds to its substrates and forms that enzyme substrate complex, the phosphate group that we have in the high energy molecule, it's going to transfer to ADP to form ATP. So just remember, for SLP to happen, we need all 3 parts. We need that glycolytic enzyme, we need the high energy molecule, and we need ADP. So moving on to SLP and the citric acid cycle, we have only 1 high energy molecule, which is succinyl-CoA. In glycolysis, we have 2, but in the citric acid cycle, we only have one. The energy in succinyl-CoA is going to come from a thioester bond, which is a carbon that is sulfur bonded. And instead of ATP, we're sometimes going to make something called GTP. It is functionally equivalent to ATP. So if you ever see GTP or GDP, just think ATP and ADP, but used in that context of the citric acid cycle. So this cycle, it's going to take place in the mitochondrial matrix, and its main goal is to produce NADH and FADH2, which are electron carriers. Those electron carriers, they're going to go to oxidative phosphorylation, which is going to make that bulk of ATP. It does not use substrate-level phosphorylation. It uses chemiosmosis. So there's only one step in the entire citric acid cycle where SLP is going to happen, and that is at step 5. We're going to have the enzyme succinyl-CoA synthetase, which is going to catalyze the reaction in which our succinyl-CoA is going to lose its CoA group. Then we're going to have GDP and an inorganic phosphate that are going to combine to form GTP. So just like glycolysis, the citric acid cycle is going to happen 2 times per glucose. So in substrate-level phosphorylation in the citric acid cycle, this enzyme substrate complex, it's going to include succinyl-CoA synthetase, our enzyme, succinyl-CoA, our high energy molecule, GDP or ADP, and then an inorganic phosphate. That thioester bond in succinyl-CoA, it provides all of the energy that we need in order to attach the phosphate group to GDP or ADP and produce GTP or ATP. So that is substrate-level phosphorylation. And just to recap, it's a direct way to make ATP or GTP. It happens in glycolysis 2 times, and it also happens in the citric acid cycle, but only 1 time. It does not require oxygen or the electron transport chain, but it does need the correct enzyme, the correct high energy molecule, and then ADP or GDP to happen. So if you found this helpful, remember to like the video and drop a comment, even if it's just to say hi. See you next time.